Okay, so today we are going to be looking at enzymes. So here is the specification again, and I'll be going through this portion here, and this portion of the specification on enzymes is quite a lot, so I'll do that in a separate video. And I'll also try and fit this in as well, and this portion will be in a separate video as well, I think, maybe. So what are enzymes? So this is the first part of the specification on enzymes. So enzymes are a biological catalyst. They speed up the rate of a chemical reaction, enabling reactions that take place in a living organism to occur fast enough for life to continue. So as they are a catalyst, they don't change during reactions, and they can be used as many times as you want. And they speed up a rate of reaction by lowering the activation energy, so the minimum amount of energy required to start a reaction. So you can imagine this activation energy being a guy who's trying to push up this rock and up a hill, and he's trying to put in energy. That energy is being put into the rock to push it up to the hill, and then that energy that's taken to push this rock up the hill is called activation energy, and then after that the rock can just go down the hill again and like speed up or whatever, but you get the idea. So it's a minimum amount of energy required to start a reaction. Now they catalyze metabolic reactions, these catalysts, and these enzyme actions can be intracellular or extracellular. So enzymes are biological catalysts. They catalyze metabolic reactions, and they can like catalyze stuff inside the cell. So you've got a cell, they can be enzymes inside the cell or outside the cell, which is what intracellular, extracellular basically means. Enzymes have an active site, and which has a specific shape, and which the shape of the active site is determined by its tertiary structure. And this specific shape is very, very important. This is what life revolves around: enzymes and their specific shape. So a certain amount of energy is required before the chemical reaction starts, so that's activation energy, and this form of energy is usually heat. So the enzymes lowers activation energy, so therefore the amount of heat energy used can be reduced, making the reaction happen at a lower temperature, and this speeds up the rate of reaction. So it's kind of like when you're sat in the car, you turn on the engine and you're waiting for the car to heat up, warm up. What a catalyst will do, it will heat up the car a lot quicker, and so you can start driving again, etc. So yeah, so here is an enthalpy diagram, an energy diagram. So don't be overwhelmed by this. So you've got this line, so this blue line right here. So that's showing you the amount of energy required for this reaction to start, and both are the same reactions, same things going on. The only difference is one is with a catalyst, one is without a catalyst, and that catalyst is the enzyme. So let's look at the one without a catalyst. So you can start here, and then suddenly you're going to go all the way up, reach about here, and then drop back down again. It's cool. Okay, so now what happens if we just change the reaction just by adding an enzyme? Well, obviously the amount of energy required to start the reaction is a lot lower, probably by 50%, a lot lower. So the time it takes for this reaction to start and this reaction to start is probably half. And yeah, that's it. That's literally it. That's all an enzyme does, is reduces the amount of energy required for this reaction to start. So as we know, enzymes are specific and only catalyze one certain type of reaction. Well, we know enzymes have a specific shape, and they keep that shape throughout the entire rest of their lives. So it's kind of like a jigsaw piece. Only one jigsaw piece can fit another jigsaw piece. You can't have one jigsaw piece fitting 12 other jigsaw pieces. So amylase, for example, is an enzyme that hydrolyzes, you should know what that means. It hydrolyzes starch, so it breaks down starch. Amylase breaks down starch. It breaks down starch to maltose, which is 2-alpha glucose molecules. Now, if we mix amylase and protein, however, nothing will happen. Well, obviously that's because the amylase doesn't have a specific shape to the protein, and this having a specific shape to the protein, there's a phrase for this, and it's called having a complementary shape. Now I'll go into a little bit more detail later on, but for now you've just got to know that enzymes are specific in nature. So I could have amylase and a bowl of custard and nothing, well, okay, maybe not. Okay, so if I have amylase and a 1p coin, nothing will happen. The amylase will do nothing to the 1p coin because it doesn't have a complementary shape or active site to the 1p coin. So main thing to take from this is the enzyme is specific in nature. So when a substrate fits into the enzyme's active site, so if we go back, so we have this enzyme here, and what fits into the shape of the enzyme is what's called a substrate. So when a substrate fits into the enzyme active site, it forms what's called an enzyme substrate complex, or you can shorten it down to ES complex. It's this that lowers the activation energy. So before, when we had a normal reaction without the enzyme, a lot of energy would be required to start it, but because we have the enzyme now, it's going to form an enzyme substrate complex, and that enzyme substrate complex is what is going to reduce the activation energy. So how does it do it? So if two substrate molecules needed to be joined together, the enzyme will hold the two substrate molecules together within the active site, minimizing repulsion between the molecules so that they can bond more easily. If the enzyme is catalyzing a breakdown reaction, so it's trying to break down a substrate into two different other molecules, fitting into the active site puts a strain on the bonds in the substrate and bends the bonds so the substrate molecule breaks up more easily. So take this for example. So you've got an enzyme, two substrate molecules fit right into the active sites of the enzyme. Adds more strain, stress, so they squeeze together, and suddenly out comes another new compound. This reaction is called an anabolic reaction. This, however, you get one substrate, goes into the active site, enzyme stresses the bonds, breaks the bonds, and out comes two new compounds. So this is called a catabolic reaction. Now, you've probably come across the lock and key model during GCSEs. Now, this model explains how the substrate fits into the enzyme in the same way that a key fits into a lock, hence the lock and key model. So the active site and the substrate have a complementary shape. So whenever you're talking about the key or lock and key model, always the active site and the substrate have a complementary shape according to this model only. So there are various other theories and models out there, but for this one, the key and lock model, you've got to say the active site and the substrate have a complementary shape, and by complementary we mean that it fits perfectly like this. It fits perfectly. Now scientists soon came to realize that this model, it's too simplistic, basic, and so new techniques reveal that proteins are not rigid structures, and we have found that various parts of an enzyme molecule move in response to a change in its environment. So Then scientists came up with a new model, not the lock and key model, like something a lot better, and this is called the induced fit model. Now, before the substrate binds to the enzyme, the substrate and the active site are not precisely complementary in shape. As the substrate binds, the active site changes shape and molds closely around the substrate. So let's take a look at this diagram here. You can see clearly those two are not complementary whatsoever. They're not precisely complementary, because you've got like this pyramid zigzaggy substrate here, and you've got this other weird substrate here, and then you've got like a curved active site. They just can't fit. And according to the key and lock model, they're meant to be complementary, but here they're not. And here we are following the induced fit model, and what happens is it comes close together and it just forms a complementary fit. And then obviously we get our product, and this is obviously an anabolic reaction. And so yeah, it's kind of if we look at the analogy of putting on a sock on our foot is not complementary precisely to the shape of the sock, and so as we put on the sock onto our foot, the sock molds around the foot, the same way here the enzyme molds around the substrate. Now, if the tertiary structure of a protein is altered in any way, so let's refer back to the primary structure, so that's a chain sequence of amino acids. Then we have a secondary structure, which is either an alpha helix or a beta pleated sheet, which has hydrogen bonds and peptide bonds. This only has peptide bonds. And then we've got the tertiary structure, which is where the secondary structure, which is either an alpha helix or a beta pleated sheet, coils even further, or like squeezes together even further, comes together a lot more and forms other new bonds like ionic and disulfide bridges. If that tertiary structure is altered in any way, the shape of the active site will change. Now, that's telling us that the active site is dependent on the tertiary structure. And so then where it has its bonds, etc., so we have an active site right here, and that active site is made because the amino acids have made certain bonds in certain places, creating this unique shape. Therefore, an enzyme substrate complex won't be made if the tertiary structure is changed in any way. So that basically means the bonds are broken, and that results in a totally different shape, meaning no enzyme substrate complex. This is really important. This is like gold dust for your exam when you're talking about enzymes and proteins. Now, the tertiary structure can be affected by pH and temperature. The primary structure is determined by a gene, so if a mutation occurs in that gene, that will change the whole enzyme, including the tertiary structure, because obviously any change in amino acid changes like the position of bonds, because this will have to go through a secondary structure, go into alpha helix, beta pleated sheet, and then go into the tertiary structure. So in the next video we will look at factors affecting enzymes.